Okay. Students, you just have to draw the iron and then 4 porphyrin rings attached to the iron for cytochromes. Write it carefully. See how I've connected them. First draw iron, 4 rings, then join them by carbon-carbon bond, covalent bonds. Okay. And here comes the first ring compound. Draw the double bond here. Should balance, no? Show the balancing. I'll first draw the functional groups here. So you'll follow CH3, methyl group will be there. And here CH, CH2. CH, CH2. Similarly, here also CH3, CH, CH2. Okay, here CH3, CH2, COOH. Similarly, here also CH3, CH2, COOH. Okay. Now continue the balancing of the double bonds. You find alternative double bonds. These alternative double bonds help in trapping the electrons. They help in trapping the electrons. Written in pink, these are the double bonds. Okay, they help in trapping the electrons. Whatever I've shown here is cytochrome B. What is it? Cytochrome B. Understood? So there will be different types of cytochromes. Okay, I was telling you about different types of cytochromes. Cytochromes are of different types based on the absorption spectrum. They vary. Cytochrome A, cytochrome B, cytochrome C. Okay. Till here, all of you wrote, this is cytochrome B. Now I will tell you the changes. If cytochrome B is like this, what will happen if the cytochrome C? First, let us discuss what is cytochrome C. This group is the second ring, CH-CH2. If this group, understand properly, if this group is replaced by, this cysteine sulfur, just draw this, CH-CH3. See here. If this group is replaced by this one, sulfur, cysteine, CH-CH3. If this group, CH-CH2 is replaced by this, then this becomes same structure, will convert into cytochrome C. Becomes what? Cytochrome C. All are writing? Yes?
Yes, sir.
Yes, sir.
Follow till here. This is cytochrome B structure. When this CH-CH2 is replaced by this group, sulfur is always attached with cysteine, CH-CH3 group, then we'll call it as what? What is it called?
Cytochrome C.
Cytochrome C. Okay. You also do the same way, just write in some color so that you just remember. Now coming to first group now. If this group CH-CH2 is replaced by different isoprene unit, that time it becomes cytochrome A. If it replaces by a different isoprene unit, I'll draw that isoprene unit down now. All of you follow till here. I can move the laptop.
Yes, sir.
Okay. Come down. Here I'll draw this isoprene unit. Just observe properly. Five peaks you get like this 1, 2, 3, 4, 5. Alternatively, put CH3 here. This is called isoprenoid, which is going to be attached in this place. In this place. Okay. Once this isoprenoid is attaching here, that becomes cytochrome A. So we'll just show that this attaches here. Sorry, here run extra 1 second. Here you show. 30%. Here I should put double bond here. Double bond will come here. Sorry, then it's correct. Then it's correct. Don't erase it, it's correct. Okay. So this double bond was mistaken. Okay. So put double bonds near CH3. This is isoprenoid unit which will replace the CH2 here. Okay. Then we'll call it as what? Cytochrome A. Cytochrome A. All of you wrote this? This will be replacing this. That time we'll call it as cytochrome A. In examination, don't worry. At least remember this one. Cytochrome B, at least you write. For your knowledge, I've just added on this also, just to understand. Got it? Should I send a picture of this?
Yes, sir.
Okay. This iron is there, no? This iron will be converting from ferric to ferrous and ferrous to ferric. Reduction, oxidation. That you should remember. Just like in iron sulfur protein, I've given you the reaction, no? What is it? Can you recollect? I'm writing this side. Just recollect. Like this. Fe2 plus, sorry, Fe3 plus to Fe2 plus. Ferric to ferrous. And ferrous to ferric. This conversion, reduction, addition of electron, removal of electron. This remember, no? This conversion, that's a basic principle of the structure. Okay. Understood all of you? Till here? Shall I send the photo of this? You'll understand the clues. All of you understood this?
Yes, sir.
Yes, sir.
Okay. So now we have learned NAD, FAD, FMN, FES, and cytochrome. All these we have learned. These all come under electron carriers. Can you name the cytochromes which are there in our ETC? What we learned? Cytochrome B, Cytochrome C1, Cytochrome C, Cytochrome A1, Cytochrome A3. Do you remember all these things in the diagram what you wrote? Got it?
Yes, sir.
Please remember, again we have B, C1, C, A1, A3, like that. Why? Because there will be differences in the absorption. This is only general structure what I have given you. People have understood that there are different types of cytochromes based on absorption spectrum. Cytos means hollow vessel. Okay. Chrome means color. That's the meaning. This was studied by a scientist by name David Killeen. David, D-A-V-I-D, David Killeen, K-E-I-L-N, L-I-N. I'll write it here. David Killeen was the first person to work on this cytochromes and all. K-E-L-I-N. K-E-L-I-E-N. David Killeen was a person who worked on cytochromes. Absorption spectrum is the main principle. If you have to learn this, how we should do, extract the cytochromes from the cell membrane and then go for study. How much wavelength they'll absorb they get absorbed. So at different wavelengths, they get absorbed. That's called absorption spectroscopy, whatever we learned, absorption spectrum. Okay. So in chemistry, we have spectroscopy chapter. Maybe I don't know whether you have it in UG, but under that, they have these studies. Chlorophyll pigments, they study. Cytochromes, they study based on absorption spectrum. Any doubts still here, children? All of you followed this? Next, we will have to study with electron transport chain is almost done now. Different, have you understood the water formation there? Partial oxygen plus 2 proton plus 2 electron gives water. Yes. In electron transport chain. Okay. And what do you find after electron transport chain? What's the last part of electron transport chain? Anyone can tell? What is the last part of electron transport chain? Complex 5. What is complex 5? What is complex 5?
ATP synthetase.
One second, I'll just take a photo. ATP synthetase. Yes. We are studying about ATP synthetase in the next session now. I'm taking a photo of this, sending it to you. One second, students. I'll send you some other pic if it is there also. I've just sent you for time being. Just have a look at it. Coming to ATP synthesis, shall I clear this now, students? All of you follow till here. You're okay with this till now?
Yes, sir.
Whatever we have learned. Okay. Now what we will do, all of you will draw this diagram, what I'm writing on the board, that is talking about the topic called oxidative phosphorylation. Very, very important for the examination. Please read properly. Oxidative phosphorylation. Okay. So what do? You should draw phospholipids. All of you draw along with me now. Draw phospholipids like this. Draw phospholipids. Draw the phospholipids like this. Draw a few lipid layers. Attachment of lipids here. Okay. This part is very important. We are studying about ATP synthetase channel that side. And here, just write roughly. Just don't mention, for example, FMN, iron sulfur protein, ubiquinone. Just write it close by, just to understand. FES, again ubiquinone, cytochrome B, cytochromes B1, cytochrome B, cytochrome C1, cytochrome C, cytochrome A1, cytochrome A3. Just like that to understand. If you want, you can label them later. These are the previous episode of this topic. Previous episode. Before oxidative phosphorylation comes, electron transport chain is associated. Now we should draw, always you should draw simply one diagram, like a long tube. Now we are going to draw the real ATP synthetase channel. Okay, see now, all of you have to draw this C rings. These are called C rings. What you're seeing here, this is called C rings. Draw like this. What are these called as? Actually, I need labeling place, so I'll write it like this. This is called C ring. C rings. Okay. Above this will be the A1 protein. See here. Draw like this. 4 units you draw. 4 units you draw. Okay. This is called A1. A1 protein. A1 protein. Then C rings. Followed by stock. Draw the stock. These all are protein subunits. Stock. Okay. And now we will write the 3 units in the stock. Observe properly. This is delta. Center one is delta. Hope it's visible for you. Delta, gamma, epsilon. Delta, gamma, epsilon. E means epsilon. I'll give you the full forms little later. Gamma, delta, epsilon. Proteins are there. Followed by this. Down will be the alpha beta sheets. What is this called? Alpha beta sheets. Means it is something like this. See my fingers here. Alpha, beta, alpha, beta. Like that. Alternative. Okay. Alpha, beta, alpha, beta. Down it's open. Down it's open. This way. Here is ATP synthetase channel. Edge will be ATP synthetase channel. It is like this. Hollow core. Got it? Understood this? Alpha sheet, beta sheet. Alpha sheet, beta sheet. Down is open. Hollow. Alpha, beta. Alpha, beta. Alpha, beta. What I've done for beta, so just see. Beta I've colored like this. Just to understand. All of you wrote till here. Is it visible?
Yes, sir.
A1. In blue, it's not visible. A1, C ring, stock, delta, gamma, epsilon, alpha, beta sheets. Okay. Now, this region, top portion, what we are learning, fill stock. It is called F0. What is it called? This region, fully the top portion. That is called what? F0, F0, like this you write. F0. And from this epsilon, delta, all those things, up to this alpha beta is called F1. F1 fragment. You have to learn this diagram properly. All of you have to learn this diagram properly. Okay, no, clear? Children, all of you understood till here?
Yes, sir.
Okay, please remember, I should show FMN ubiquinons with pumping of protons. All of it write that now. Proton pumping. See here, like this. 2 proton. This one will give 2 proton. This one will give 2 proton. Like that you should write. Proton pumping. Okay. When protons will be more, they'll create what? PMF, proton motive force. They'll create proton motive force. That proton motive force will influence the A1 protein. It influences the A1 protein. Just to make you understand, remember These all protein units are there, no? These all protein units. They're associated together. They're bound together. Down is the ATP synthetase enzyme. Down is the ATP synthetase enzyme. ATP synthesis enzyme. Enzyme means it will have substrates, no? Can you name the substrates, any one of you? ATP synthetase is an enzyme. Whatever drawn here is called what? ATP synthetase channel. I don't have place, I'll draw it down. I mean, I'll name it down. Just see here. I'll name it down. ATP, you don't write it just below the diagram. Go away from the diagram and write a few points. ATP synthetase channel. ATP synthetase channel is whatever I've drawn on the right hand side. It will have what components? F0 and F1 fragment. It will have F0 and F1 fragment. In that F0, it is having A1 protein, C rings, stalk, all that will be there. In F1 fragment, it will have gamma, delta, epsilon, alpha, beta sheets. All this will be mentioned. You have to explain it. I'll give you notes. You have to explain ATP synthesis channel. Those proteins are called as Fernandez-Moraz units. They worked on this. Fernandez-Moraz units. Okay. They're also called as Parsons. P-A-R-S-O-N-S. Parsons. Okay. So Fernandez-Moraz units or Parsons. Or sometimes they're also called as oxysomes. They're called oxysomes. These proteins are there, no? These proteins. These proteins, actually, these proteins are very hollow. Like this, they are hollow cylinders. Sorry. These proteins are hollow cylinders. Okay. Between one protein and another protein, they are associated like this, something like this. Imagine. Okay. The gap between each protein is 100 angstroms. The distance between each protein is 100 angstroms. It's hollow cylinders of proteins. Parsons or Moraz, whatever I was telling you, oxysomes. 100 angstroms difference will be there for each. Got it? It's hollow cylinders. To tell you one more information, F0 fragment will be 50 angstroms in size. F1 fragment will be double of that, 100 angstroms in size. Clear? All of you understood some points on this?
Sir, can you repeat this once more, sir?
The protein units are called as Parsons or Moraz, Fernandez-Moraz units or oxysomes. Each protein will be cylindrical and elongated like this, hollow cylinders. Okay. The distance between each protein will be how much? 100 angstroms. Okay. And the whole size of the F0 fragment will be 50 angstroms. And that of F1 will be 100 angstroms. I'll send you some notes on this afterwards. I'll be sending you, don't worry about it. Okay, so these, what do you say? I mean to say these F0 and F1 proteins are like this. They're associated together. Whenever there is, what do you say? Whenever there is a proton affecting these units, they will bring a change. Now I'm talking about synthesis of ATP. Till now I told you only ATP synthetase channel. Other name for this ATP synthesis channel is proton channel. Can I erase this portion? This one I'll erase. You followed it? The points I'm telling, these points. How are the proteins, all those things. Okay.
Yes, sir.
Yes, sir.
I'm just trying to put some keywords for you to follow here. Just note these points and continue the next class. 4 minutes is there. 3 minutes. Please take the screenshot. Ma attendance, Meenakshi.
So I've already taken it, sir.
Okay. See here. You have to learn some concepts here. Chemi-osmotic theory. You have to learn chemi-osmotic theory that was given by a biochemist. Is it clear? The board is clear?
Yes, sir.
Biochemist by name Peter Mitchell. He tells about chemi-osmotic theory, which I'll discuss in the coming class. Chemi-osmotic theory was given by Peter Mitchell. Okay. He tells about pH variations. He tells about pH variations and talks about osmoregulation. Can anyone try to guess this, what I'm trying to say? pH variation, osmoregulation, can you connect it? It's all, I'll give you the clue. It relates to protons. If you have done it, I'll remove this board. So you'll see the back, the other board. Have you written this? Keywords, chemi-osmotic theory, pH variation, osmoregulation. Have you written this?
Yes, sir.
Yes, sir.
Just see the board now. I've given the clue to you now. See the board now. Protons are there. Tell me now. Try to guess what he's trying to explain. pH variation. Word pH variation. All of you make a note of it. Whenever protons are pumped inside this membrane here, you find proton motive force. No, the pH will be actually very less. Note down pH 4. No place for me. I'm just writing it here. pH 4 will be there in the membrane space between cell wall and cell membrane. If it's bacteria, it is mitochondria, outer membrane and inner membrane. Between this space is called intermembrane space. There you can find pH variation, pH 4. Automatically, the pH will be more in case of cytoplasm, pH 8 or pH 9. I'll tell you fast. We have 1 minute left. Okay. This pH variation he tells about. There should be osmoregulation. What will the protons do? They will enter the proton ATP synthesis channel for osmoregulation. Protons will come into this and then they'll come in back into the cytoplasm to regulate the pH. So hydrogen ion concentration plays a very important role here. pH 4, pH 8 is called pH variation. They need to show osmoregulation, so they'll enter the ATP synthesis channel. Otherwise, it's called as proton channel. ATP synthesis channel is also called as proton channel for bringing osmoregulation. In the process, ATP synthesis will happen. That I will continue in the next class. I have less than a minute. Please make a note of these points, what I told you. Osmoregulation means here pH, hydrogen ion regulation. All are clear till here? Yes, students?
Yes, sir.
Okay. In the coming class, we will discuss that. Means I'm talk only on ATP synthetase enzyme. And then we'll continue with the next topics. Okay? All of you take care. We'll meet. It will de-link anytime. It's just a few seconds. I'll send the diagram for this. You want the diagram?
Yes, sir.
I'll send you this diagram and some points also. Okay. Please tell other students, only 30 are there. Remaining students are absent here. Okay. This is an important topic.